I'm back on the mark again. Okay. Okay, so we'll go into Chapter 9, Biotechnology and Recombinant DNA. Some technical jargon here, but again, primarily, just like the last chapter, some definitions I want you to know and appreciate. Some of the intricacies of biotechnology, recombinant DNA, not necessary for you to know, but again, just an appreciation for how this type of technology is now being applied in our environment. Biotechnology basically is the use of microorganisms, cells, or cell components to make a particular product. Foods, antibiotics, vitamins, enzymes, all now produced utilizing combinations of biotechnology and recombinant DNA. Recombinant DNA technology is basically the insertion or the modification of a gene to produce a desired protein. And this can be done in mass quantities now. And we'll talk about a little bit about how vaccines are produced utilizing this, certain medications such as insulin produced utilizing this mechanism. Again, we've used the term recombination prior in the previous chapter that we were talking about the reshuffling of genes between two DNA molecules forming recombinant DNA occurs naturally in microorganisms. It's also possible to manipulate this DNA artificially by combining genes from two different sources, even in the biological system, the human system, which we're familiar with, and utilizing that technology also in bacteria as well. Artificial gene manipulation is known as genetic engineering, and the term biotechnology usually means that there's an industrial use associated with this genetically engineered microorganism. Now, the gene of interest is first inserted into what's referred to as a vector DNA or a cloning vector. The DNA molecule used as a carrier must be self-replicating. So what's used a lot are plasmids and viral genome. Recombinant vector DNA must enter into a cell where it can multiply, forms a new clone genetically identical to the previous cell. The gene itself may be then the desired product, or it may be the protein product. So it may not be the cell itself, it may be the products or byproducts of those particular types of cells that are expressed by the gene that you want, say, in an industrial application. To create recombinant DNA by cutting off the DNA or by cutting off the donor DNA and inserting a given fragment or small replicating molecule or a vector, which under certain conditions will amplify the fragment along with itself, resulting in a molecule or a clone of an inserted DNA molecule. The vector molecule with their inserts are then called recombinant DNA because they represent a combination of the DNA from the donor genome with the vector DNA from a completely different source. Again, this can be bacterial, plasmids, and or viruses. Now, there are some tools or instruments that have to be used in biotechnology. These are as follows, one of which would be a restriction enzyme. This is a special DNA cutting enzyme. They're used in genetic engineering. They basically have a natural function in protecting bacteria from attack by phages and hydrolyzing their DNA. And again, make sure you look at the, I know some of you have already done the lab for this week, but it talks about phages in significant detail, so make sure you look at that little media that goes with that. The DNA of a bacteria is protected from the enzyme by the addition of a functional group. The functional group is a methyl group. We see this on some cytosine methylates. And basically, this will help, the addition of this will help protect the enzyme. The enzyme then recognizes and cuts into the two DNA strands. Sorry, I got a typo there. Cuts into the two DNA strands. The cuts are not directly opposite of each other. Stretches the single DNA at the ends of the DNA fragment. These ends of the DNA fragment referred to as the sticky end. They stick to the complementary stretches of the single-stranded DNA by base pairing. If two fragments of DNA from different sources have been produced by the same restriction enzyme, then the set of sticky ends can then be spliced and recombined easily. A DNA ligase then links these DNA pieces together. This is an illustration out of your book that basically shows the steps of recombination. And again, this is not necessarily, I'm not putting this up for you to have to restate these steps to me, but I just want you to have an overall appreciation of the process that's involved. These are some pharmaceutical products that are now currently being used and that utilize genetic engineering. Alpha interferon, when we talk about immunity later on, and for those of you in ANP2, we talk about the lymphatic system and the immune system, and we'll talk about interferon a lot. It's an important factor in the immune system. If you can produce that in a mass quantity to be able to administer in a therapeutic level to in a clinical setting to individuals, then you can see the obvious advantages to being able to produce large portions or large amounts of interferon, and you can do that through genetic engineering. Antitrypsin, another drug that you'll see used a lot is erythropoietin, EPO, that you can see further down the list on this table. It's used in the treatment of anemia. It's typically only produced in the kidneys. However, again, utilizing being able to manipulate the genes in certain types of organisms, you're able to produce this in mass quantities in a cultured cell. Factor VIII we see used in hemophilia, bleeding disorder. Human growth hormone corrects growth deficits in children, utilizes E. coli as the genetically engineered structure to be able to produce that in mass quantities. And human insulin, again, therapy for diabetes, I've already alluded to that earlier. It's produced as well utilizing E. coli. So you can see there are significant applications to using genetically or genetic engineering. We really, if you remember last flu season, there was a shortfall of influenza vaccine, basically because it takes a period of time to manipulate. First you have to grow these organisms, but you have to grow genetically engineered microorganisms over a period of time. And so for this genetic manipulation to take place, to make sure you're growing out the right strain of a given type of virus or bacteria, it takes a period of time. And so if there's a shortage this year, it's next year before we can recover from that, because it takes a period of time for this genetic manipulation to take place. But you can see again there's a number of different drugs that are utilized. Again, when you see an industrial application of genetic engineering, that's when we look at it as biotechnology or an application of biotechnology. Selections and mutations. Selection, basically a culture, a naturally occurring microbe to produce a desired product. The mutation we already talked about, mutagens, which cause particular mutations that might result in a microbe with a desirable trait. Site-directed mutagenesis basically is a change in a specific DNA code to change a protein. Remember, it's the synthesis of proteins that we're looking for as the end result of DNA manipulation. And then select and culture microbes with a desired mutation. Restriction enzymes, again, are used. One of the tools that's used in recombinant DNA. Restriction enzymes will cut off specific sequences of DNA, destroy bacteriophages' DNA in bacterial cells. It cannot digest the host DNA because it's methylated. As that functional group, that methyl group, methylates the cytosines that prevents it from being destroyed. What are vectors? Again, the vectors, they carry new DNA as desired cells. The shuttle vectors basically can exist in different types of species. Plasmas and viruses are basically used as vectors. Now, don't confuse this definition of vector with what we use as vector later on in the semester. Later on in the semester, we talk about blood-borne diseases and microorganisms that can cause different diseases. There are various vectors. A mosquito is a primary vector that's allowed to be able to carry disease from one organism to another. And so a little bit different than what we talk about here. We talk about plasmas and viruses being vectors. Now, host cells can be induced to take up particular plasmids. Plasmids, again, used as vectors. the diagram, previous diagram basically showed you the DNA fragment of a particular vector. Plasmids can exist in several species as shuttle vectors, in other words, transmitting these genetic information from one site to another. Plasmids is an independent, circular, self-replicating DNA molecule that carries only a few genes. So just a little bit of genetic information is transmitted there. The number of plasmids in a cell generally remains constant from generation to generation. Plasmids basically are autonomous molecules that exist in cells, extrachromosomal genomes, although some plasmids can be inserted into bacterial chromosomes where they can become permanent part of the bacterial genome. They manipulate and isolate using bacteria. They can also be integrated into mammalian genomes, so we can see their application there. They can confer mammalian cells or whatever genetic functionality they have to carry. It gives you the ability to introduce genes into a given organism by using a bacteria to amplify or to make greater, to amplify the hybrid gene that's created in the test tube. This tiny but what's referred to as a mighty plasmid, because it carries enough information to be able to carry this amplification out, is the basis of recombinant DNA technology. There's been a lot of recent developments utilizing methods to be able to analyze or use an analysis to be able to determine the DNA constituents of various types of organisms. One of these types of methods is referred to as the PCR, or the polymerase chain reaction. Basically, what you do is you start off with just a particular piece of a gene in the DNA, and then this PCR, this polymerase chain reaction, will make multiple billions of copies in a very short period of time. In this reaction, a target piece of DNA is basically heated. It's separated from the original DNA strand, which serves as a template. Remember, if you heat a protein, if you change the pH, you'll denature it. And so in denaturing it, the DNA, the double helix is coiled, and then it's supercoiled. But if you heat it, you can denature it. And so basically, in heating this targeted piece of DNA, it will separate the DNA strands. Then the DNA polymerase, which you've heard of before, it's an enzyme which basically forms DNA by linking nucleotides. It also proofreads the complementary base pairs there to make sure no errors are made. Then each newly synthesized piece of DNA serves in turn as a template for more new DNA. So if you have a biological sample, a small amount of DNA, you want to work with this DNA, and because you only have a small amount and you need more of it, then you would want to use this PCR, this polymerase chain reaction, to amplify or to make more pieces of the DNA. And so that's what the PCR does. It does an amplification process, takes a small amount of DNA, changes it into a larger amount. It's quickly, easy. Billions of copies are made in a very short period of time. Large amounts of DNA mean more accurate, reliable results for you to use in later techniques. So this is a big thing that's used in labs that are doing research, because if you only have a small amount of material on a particular substance that you want to do a lot of studies on. A good example of that are these viruses that, the smallpox virus, for example. We don't want a lot of smallpox around. But in a controlled research laboratory, they want to do more studies, say, on smallpox, to find out if there are different drugs that you'd be able to treat with that. You want to find out what particular enzymes are secreted, how different chemical substances work on that enzyme. Well, you don't have much of the virus, so you use a PCR, and you can create millions of copies of that particular virus. And then you can do, so now you have more of a sample to be able to do studies on. Smallpox may or may not be a good example to use in this, because you don't want a lot of that agent out. Recently, you may have heard about a virus, a particular virus strain that they sent around to labs, research labs all over the country. These were high-level labs that were being tested on their ability to identify particular types of organisms. And they used the polymerase chain reaction to recreate a significant number of the virus, and then they sent it around to the labs. They got a lot of bad press because this was the virus they used caused a lot of deaths, you know, many years ago. And so they were like, well, if you're going to replicate a virus, why did you have to pick one that was so virulent? But they got all the samples back. Nobody died from it. But it just shows you the technology that's out there right now to be able to do it. The PCR technique was developed by a Nobel Prize winner, Kerry Mullis, in 1984. Important to know, you won a Nobel Prize, somebody should talk about you at some point, so I'm just trying to give him his respect. That's all you need to know about that. I'm not going to test you on his name. PCR rates versus cancer cells. Now, cancer cells kind of do the same thing. I mean, they grow rapidly and out of control as well. There's an amplification process in DNA with the cancer cells as well. But amplification of DNA by PCR is even faster than what you see in cancer cells. And those of you who may have some clinical experience with cancer recognizes how fast that certain cancers can grow. But the PCR amplification is much faster than that. We know it is high reproductive rates. PCR can make one of a single strand of DNA into billions of copies in a few hours. A cancer cell would take a month or so to do the same thing. So it gives you some indication of how fast this process can take place. If you want to make multiple copies of a piece of DNA enzymatically, again, you use this polymerase chain reaction. Polymerase ASE tells you that it's an enzyme involved in the process. Clones DNA for recombination, amplifies DNA to detectable levels. The sequence of DNA gives an adequate and a correct sequence. Diagnose genetic diseases. It can be used in that. How does it diagnose genetic diseases? Well, the polymerase chain reaction in a laboratory setting, they can identify the particular nucleotide-based sequences based upon their imaging. They can either put it through a particular type of scan, computerized scan, to be able to identify the sequencing that's there, and it gives a specific ID on a particular type of microorganism or a genetic disease. For HIV, for example, it helps in detecting the viral pathogen because, again, there is a specific reaction that we see in the polymerase chain. So not only can you use it in amplifying DNA to detectable levels to do in research studies, but you also can sequence the DNA utilizing this technique, and then you can also diagnose genetic diseases and or detect particular pathogens. So it's a highly specific study that's used now in many laboratory settings. Okay, now technique of genetic engineering, injecting or inserting foreign DNA cells or foreign DNA material into cells. Plasmid vectors can be inserted into many different cells, chemical treatments that make transformation impossible. Such cells are called competent, or they have this ability to take up external DNA. In the technique, we see the walls of these cells are enzymatically removed. They exchange DNA for what's referred to as a protoplast fusion. There are different ways to make this mechanism work. You can use polyethylene glycol, which can be used to improve the efficiency of this technique. You transferring of DNA into this manner. It's also enhanced by utilizing an electric field that forms little small pores, and through these pores, information can be injected through this protoplast membrane. That process is referred to as electroporation. Foreign DNA can then be introduced into plant cells, coating microscopic particles of tungsten with the DNA, and you can fire this through a particular type of gun, and I think it's a picture of that gun in your textbook, and if not, I think I might have one on here as well. But it basically has they refer to it as a gene gun, where basically you're going to fire this piece of DNA through a cell wall, particularly of a plant, utilizing a gene gun. DNA can then be introduced in the cell through minute glass micropipettes. This is a very delicate procedure that has to be done, and that's referred to as microinjection. Yeah, here's a picture of the gene gun. The gene gun basically utilizes low-pressure helium pulse, delivers particles. In this case, this particular particle is coated with gold. Previous slide we said tungsten, but you can utilize any one of these. It's virtually inserted into a targeted cell or tissue. Particle carry the DNA so that you don't have to remove the cell or the tissue in order to transform the cell. It's a form of gene therapy, genetic immunization, DNA vaccinations can be achieved through this way. So if you want to inject DNA into a cell for recombinant purposes, transformation, electroporation, or protoplast infusion are the three different ways that you'd be able to carry that out. And this is just an illustration showing you the micro-injection using a pipette. You can see how delicate of a procedure that is to be able to do that. Obviously, you have to do it microscopically to be able to do it. And then we talked about the gene gun. It's amazing what people do in labs. I mean, to me, that's just, not that I'm into that, but it's just, that's an amazing process. And then obtaining DNA. So where do you get the DNA from that you want to do this on? Well, there are gene libraries that are made of pieces of an entire genome that is stored in plasmid or phages. And basically, DNA is made from messenger DNA utilizing a specific enzyme called reverse transcriptase. Synthetic DNA is basically made by DNA synthesis or DNA synthesis machine. Genes from a particular organism are isolated by cutting up the entire genome with these restriction enzymes that we talked about earlier. As you splice them, there are many possible vectors then that are utilized to introduce genes into bacterial cells. Then these collections of bacterial clones containing different DNA fragments are known as the gene library. And then you can basically go there and pull out, just like you go to the library and pull out a book, you basically pull out the genetic information that you need, utilize this in a different organism to be able to make multiple copies of it or to amplify it. And if we're doing this process in eukaryotic organisms, cloning genes from eukaryotic organisms generally require the removal of stretches of DNA, intron stretches of DNA, that do not code for specific proteins. You want those out because they basically just take up space. This is done by splicing. The introns are removed when the RNA transcripts of a gene are converted into messenger RNA. This remains. What's remained are referred to as exons or stretches of DNA that code for specific proteins. And this is basically what you want. You use an artificial gene that contains only exons can be used with an enzyme, the reverse transcriptase, and then this will synthesize complementary DNA sequences from the RNA. Most of these methods are used in obtaining eukaryotic genes, especially